Hi, my name is Danny Smith, and for my project, I chose primary congenital hypothyroidism as my disorder. Excuse me. So, some background information on this particular disorder is that it is an endocrine disorder, and it is characterized by an abnormally sized thyroid gland, or actually, I should say, abnormal thyroid hormone production. So the thyroid gland is a butterfly-shaped gland located in the lower region of the neck. A normal thyroid gland, or a normal-sized gland, produces a normal level of thyroid hormones, and these hormones are rich in iodine, and they are necessary for healthy brain development, um, as well as growth and metabolism in newborns. A shortage in these particular hormones causes the disorder, and the shortage of hormone production is caused by, like I mentioned earlier, an irregularly sized thyroid gland. Another cause might be that the chemical synthesis for which the hormone is produced, may have some kind of problem, some chemical problem within that process, causing a lower production of hormones. So, in this case, you might have a normally sized thyroid, but because of an error in the synthesis, you're going to have a lower production of that thyroid hormone. So, as for the history, I could not find any information as for when this particular disorder was first discovered or when it was first reported. I looked and looked and looked, and I could not find anything on it. So, I apologize for that. I don't have anything to report on that in my presentation. As for the prevalence, it affects 1 in 2,000 to 4,000 births in the United States every year. A larger prevalence is known in less developed countries, and the primary reason for this is that there are a lack of newborn screening tests available in many underdeveloped countries, and since these newborn tests or screenings are what first indicates or identifies that a baby has the disorder, there is a larger prevalence in countries for which they don't have this particular screening test. Another point is that it affects twice as many females as it does males, and according to the NCBI website, the reason behind this is unknown. So some symptoms, I should say, they vary between infants. Some babies may have more sleepiness, they may sleep more, they may be less active. Some babies have difficulty feeding and experience constipation. But a lot of infants have no symptoms at all, and in fact, a lot of a lot of babies show no physical signs of the disorder, which is why early detection is so important. If untreated, hypothyroidism is known to cause slower growth as well as intellectual disability, which is very severe. If treated within 2 weeks of birth, however, newborns usually develop normally without any symptoms of the disorder. So, the responsible gene and chromosomal location. Hypothyroidism results from one of many possible gene mutations. So, here's a list of all the genes known to be associated with thyroid production. And it just takes one mutation in any one of these genes to cause that hypothyroidism disorder. One thing to note, though, is that the disorder is largely sporadic, which means it's not always inherited. In fact, only 2 to 5% of all hypothyroidism cases are inherited, and this is usually associated with the PAX8 gene or paired box 8 gene. This particular gene is located on chromosome 15, and its specific locus is 15q 25.3 through 26.1. So for the mode of inheritance, this disorder is known to be associated with both an autosomal recessive inheritance type and autosomal dominant. However, hypothyroidism is usually associated with an autosomal recessive inheritance type. This is the most common. So I just want to mention, though, that dominant, in the case of a dominant disorder, this results from mutations in the thyroid synthesis hormone gene or the DUOX2 gene. And with dominant disorders, all dominant disorders, it only takes one dominant allele to cause the disorder. However, it is much less common than autosomal recessive. So for autosomal recessive, it takes 2 recessive alleles in order to have the disorder in your phenotype or genotype. And Since this is the most common, I'm going to continue to use this as the template for the rest of my presentation. It only takes one parent to inherit this disease or disorder. However, the probability of offspring receiving the disorder depends on the second parent, whether or not they're a carrier of it or are afflicted of it. It, indicates or dictates, I should say, the percentage of probability that the offspring might get it. This disorder can also occur from mutations in formation of sex cells, and in these cases there might not be a family history of the disorder. So for one of our project template assignments, we had 4 possible inheritance scenarios that we went through. I'll just quickly go through that. So scenario number 1, both parents are afflicted with the disorder, and since it's recessive, both parents are going to be homozygous recessive, and all the offspring are going to be afflicted with the disorder because they will also be homozygous recessive. Scenario 2, there's one afflicted parent and one unafflicted parent, and they are not a carrier. So that means the second parent will be homozygous dominant. The result will be that there will be no chance or no probability of offspring being afflicted. However, all offspring will be a carrier since they all have one recessive allele for the disorder. Scenario number 3, there's one afflicted parent and there is one carrier. The other parent does not have the disorder, but they do have one recessive allele for which they carry the disorder. So the results, when we plug it into our Punnett square, we'll see that we have a 50% probability that offspring are afflicted here, and then a 50% probability that offspring are carriers but are unafflicted here. Finally, scenario number 4, neither parent is afflicted nor a carrier. So in this case, both parents are homozygous dominant, and all offspring will be homozygous dominant. So there is no chance or no probability that the offspring will inherit the disorder or be a carrier. So this is the pedigree I did for another project assignment. And the only thing I wanted to point out here is that for a autosomal recessive disorder, the disorder can pass generations. So if you'll notice here, with looks like Penelope and Dimitri, they do not have the disorder, but their child does, and this is because they are heterozygous and they passed they each had a recessive allele, lowercase t, that they passed on to one of their children. So the gene responsible for the disorder is called the paired box gene 8, and this codes for a nuclear protein involved in follicular thyroid cell development, and it functions in expression of specific thyroid genes. PAX8 activates genes during embryonic development, and one thing to notice or to note is that not only does it affect the thyroid, but the PAX8 gene also kind of dictates the activity of genes related to kidney formation. So a mutation in the PAX8 gene can result in hypothyroidism, but it can also result in possible disorder related to the kidney. So this is just simply the nucleotide sequence for a normal PAX8 protein. I'm not going to read it all. You can look at that. Also have the accession number in case you want to track this down and see it for yourself. It's listed here. One thing to note is just that it's right under 1,500 base pairs long. However, not all of those base pairs actually code for the protein. So the protein is only coded with a coding sequence which is right under 1,200 base pairs, and it starts at 161 and ends at nucleotide number 1,357. And then we know that each amino acid is the result of a sequence of 3 nucleotides or triplets. So if you take the number of nucleotides in the sequence from the previous slide and divide it by 3, that's how many amino acids are used in developing this protein, which is 398 amino acids. Here's the accession number if you want to access that, and then this is the entire sequence of amino acids. So there are more than 15 known mutations associated with the PAX8 gene or protein in which the hypothyroidism is associated with. And these are usually due to a frameshift mutation, which is one nucleotide deleted or inserted that changes the shift of reading, or the frame of reading for which the DNA is coded for. There are only 3 specific isoforms provided by the NCBI website, and that is PAX8C, D, and E. D is a variant missing 2 alternative exons, and then E is a variant missing 3 alternative exons. All of these variants result in a frame shift mutation, and all of them cause an early stop codon. And this early stop codon is the explanation for why the thyroid gland is often smaller in size or missing altogether. It's because the coding for it has not, for the protein, has not finished. So that results in a deformed thyroid gland. So the impact of mutations on the structure/function. As mentioned earlier, a normal PAX8 protein regulates the activity of specific genes that are related to development of the thyroid gland. So a mutated PAX8 protein will not regulate that as successfully. And the result of that is an abnormally small PAX8 protein due to a mutation in the PAX8 gene. And so an abnormally small PAX8 protein is, the consequence of that is, it's a consequence of disrupted protein production. Again, a smaller thyroid gland produces less hormones. And then these are some of the types of congenital hypothyroidism associated with mutations in this particular gene. Thyroid dysgenesis is the most common type, however, and this includes all cases related to problems of thyroid gland development due to mutations. So there are some available genetic testing options. However, it's limited to newborn screenings, or I'm sure you could, do some later on in life. But the problem with that is that if you're not diagnosed and treated immediately as your thyroid gland develops throughout life, and you're producing less and less hormones, it's going to potentially cause mental retardation. And so because of that, newborn screening is really the only option to treat CHT. There are 2 types of newborn screening tests, though. The most effective and most common is the TSH test, blood spot thyroid stimulating hormone test. This test is more specific in diagnosis of the disorder. There's another test, though, and this is the T4 test, and this is more beneficial for detection of a rare form of hypothyroidism, which you can read there. I'm not going to try to pronounce it because I'll probably mess it up. However, this is not as specific with high frequency of false positives in premature infants. So some more facts on genetic testing is that newborn screening is mandatory by the government in, I believe, all states. Some sources have said some states or most states, but the textbook says that all states require this. Hypothyroidism is one of the 4 most recommended conditions to screen for. The other 3 include PKC, I believe it is, and cystic fibrosis. And there's one more. I can't remember the name of it. It's just a fun fact. Most screenings are performed 24 hours after the birth of the infant, though. And this is to account for a surge, a TSH surge, neonatal TSH surge. So after the levels iron out, the test is performed, and this reduces risk for false positives. Diagnosed infants do need to be reevaluated after 3 years of birth, and this is because it takes time for the thyroid gland to fully develop, and so they may not have it after being treated for the first 3 years of life. Their thyroid might function properly, so they do need to be reevaluated for whether or not they need treatment after 3 years of age. Early detection is extremely important, though. Infants do not normally show physical signs of the disorder, and consequently the disorder can be overlooked. A lack of treatment, again, can cause mental retardation. Here's a few companies offering the test in the United States. There's some controversy associated with newborn screenings, and that is because some hospitals have been known to sell infant blood samples, and lawsuits have been filed due to unconsented use of infants' blood samples in scientific research. It's sometimes considered an invasion of private property since it's DNA. That's all I have. Here's my reference slides. I'm out of time. I wish everybody the best of luck in the course, and thank you for your time.